Just a heads up, we are recording this webinar because we would like to make sure that we can share it afterward. And my name is Nicole Martin. I am an associate director. I'm the associate director of the Milk Quality Improvement Program at Cornell University. I'm also an assistant research professor in the Department of Food Science. And my area of research is dairy foods microbiology. I work from the farm through processing and even at the consumer level. And we are partnering here with NOFA New York today to give you an overview of a recent project that was funded by the USDA Organic Research and Extension Initiative program. This has been a 5-year project, and we're just finishing up the work on this project. So we are sharing the results that are relevant to organic dairy producers. And as you can tell from the title of this presentation, we're talking about udder hair removal today and spore forming bacteria. So I'm going to go ahead and get started. It's a small group of us today, and that's always kind of nice. It's nice to have a lot of people, but it's also nice to have a small group and we can have a conversation at the end. So if you have questions along the way, go ahead and feel free to put them in the chat or just unmute when we get to the end, and we'll have time to have a quick discussion. So, all right. I think get started. So just so we're all on the same page, and I'm just going to hide my controls here. There we go. So we're all on the same page about what spores are, because those are the bacteria that we're studying in this project. These are bacterial spores, not fungal spores. You might have heard of spores before, or maybe you've kind of run into them before in various testing that you've had done on your raw milk or in your dairy. These are really unique structures in the bacterial world. So what happens is we have a bacterial cell, and that cell can sense when there are stressful environmental conditions. So things like low moisture, low nutrients, they can sense when that's happening. And they go through this series of steps to create this very resistant structure that we call an endospore. And you can see here in this image that I have on this slide, the structure in the center that's labeled endospore. This is a series of layers of protein, and that protein surrounds the genetic information in this cell, and it allows that cell to survive what would otherwise be a lethal conditions for those cells. So it allows the cell to survive very high temperatures like we would experience during pasteurization, or very dry conditions like those that are found in dry dairy products, also radiation and sanitizers. So lots of very dairy relevant environments. These structures allow the organism to survive under those conditions. And then when the conditions are favorable again, when the conditions return to favorable conditions, these organisms can then start to grow again. And that's exactly what happens in dairy products. We get these endospores in the milk at the farm level. So they're transfer from the environment of the farm where they're found naturally. So they're naturally found in water and in soil and in plant material. And they are able to transfer into the milk because of different milking practices that happen. And then they survive these conditions that we use for pasteurization and cheese making and a variety of different dairy product manufacturing. And then they grow in the product after that processing, and they cause spoilage predominantly. There are some that can cause illness, so pathogens that can cause illness, but mostly these are considered a spoilage organism. Okay, sorry. This is a a time-lapse video of this process of a spore turning back into a vegetative cell. And you can see that there are a number of spores here. Those are those bright spots. And as the time-lapse video goes on, you can see some of them turning dark and then starting to elongate. So this is a process that we call germination. This is essentially just that dormant spore turning back into a metabolically active vegetative cell. And then that's the point where these organisms start to grow and produce enzymes and produce metabolites that can cause spoilage in the dairy product. And there are a couple of different dairy products that spore forming bacteria cause spoilage in. And the first is fluid milk. So fluid milk in the US is mostly pasteurized using high temperature short time pasteurization. But we have a little bit of what we call vat pasteurized milk. So that's a lower temperature for a longer time than high temperature short time pasteurization. But we see spoilage from spore forming bacteria in both of those types of milk. So the predominant organisms that we are concerned with from a fluid milk perspective are things like Pennybacillus and Viridibacillus and Bacillus stephanensis. Here I have a plot that just shows the growth pattern of these cyclotolerant spore-forming bacteria in fluid milk. And you can see here on the x-axis, we have number of days post-pasteurization. So again, we expect about 14 to 21 days of shelf life on a high temperature short time pasteurized milk. And then just the total count of bacteria here on the y-axis. And then we have this broken down into two different types of groups of bacteria, because there are two groups in fluid milk that cause most of the spoilage. The first is Pseudomonas, and you might have heard of Pseudomonas before. It grows really rapidly at low temperatures, and it is responsible for about 50% of the spoilage of fluid milk in the US. And when we look at the growth pattern of Pseudomonas, it grows really quickly. So really, we're looking at very fast spoilage with Pseudomonas. So it's this top line here. We reach spoilage levels 7 to 10 days after pasteurization. With the spore formers, on the other hand, they don't even grow for the first 10 days of after pasteurization. But then after they start growing, they grow pretty rapidly. And by about 17 days after pasteurization, they reach spoilage levels, which is about 1 million cells per milliliter of milk. That's about the time when they start creating some defects in the milk and the quality starts to degrade. Where these spore-forming bacteria are really important is for processors who are trying to extend shelf life. And they might want to extend shelf life for a number of different reasons. It could be because they want to reach new markets, right? If I have a sort of an existing market and I want to go beyond that, I might need a day or two extra of shelf life to make sure that my product can reach that market and get sold, and the consumer doesn't experience a short shelf life on their end, right? Or it could be they just want their consumer to have a very consistent positive experience with their product, and so they want the shelf life to be a little longer so that they have confidence that the product is going to taste great for that entire shelf life. But for whatever reason, they want to extend shelf life. These spore formers are really the organisms that prevent them from being able to extend that shelf life, as long as they don't have problems with Pseudomonas, which is introduced after pasteurization. Here's a video that a student in our lab made of a spore former inoculated into milk. So the defect that we see here is called sweet curdling. So you can see it's pretty chunky, right? Essentially what happens is when this bacteria grows, it produces enzymes that break down the proteins in the milk, and then they coagulate together, and it causes essentially curds in the milk. And it's called sweet curdling because there's no acid development. So unlike, lactic acid bacteria that produce curds and whey, they do that through acid development. The This bacteria does it through enzyme development. So it's a little bit different of a mechanism, but it kind of comes out looking about the same, except this probably tastes a lot worse because it also produces a lot of bitter peptides, very small proteins that can be bitter. So obviously we don't want consumers to experience this type of milk. So we want to make sure we're controlling these psychotolerant spores. And psychotolerant is just a word that means they can grow at low temperatures, right? They're tolerant of cold temperatures. Another dairy relevant spore forming bacteria is what we call anaerobic butyric acid bacteria. And these are spore formers that only grow in the absence of oxygen, right? So bacteria either able to grow under the absence of oxygen or the presence of oxygen. And these ones only grow when there's no oxygen. And so they happen to find those conditions in the interior of cheese, right? There's no oxygen there, and so they're able to grow. And these bacteria, Clostridium tyrobutyricum is sort of the classic example of an anaerobic butyric acid bacteria that we find causing cheese defects. And I have a photo here of some controlled cheeses on the bottom here, and then 2 cheeses that were inoculated with this spore-forming bacteria, Clostridium tyrobutyricum. And you can see that the ones that had this bacteria have a lot of open holes in the cheese. And this is what this bacteria does, is it produces a lot of gas late into the aging process. So we call it late blowing because it happens 60 to 90 days after the initial cheese making. And you can imagine that if you're a cheese maker and you're making a cheese that this bacteria can grow in, and you've put all of the work into manufacturing the cheese, into aging the cheese, and then 60 days down the road you end up with this spoilage. This cheese is essentially just not saleable at this point. There's no way to salvage it because along with all of this gas production, you get a lot of butyric acid development, and that is a very unpleasant acid, and you wouldn't be able to even use this in sort of like a shredded cheese or a spray cheese or any kind of cheese byproduct because of these organic acids that are produced. So we've known about these spores for a really long time in the dairy industry. And in fact, we've known that there's an association between poor quality silage and these butyric acid bacterial spores. So much so that in certain countries where they manufacture a lot of cheeses that are susceptible to this defect, cows can't be fed silage because of this very strong connection between silage quality and the presence of these spores in the milk. In other countries like the Netherlands, they make, obviously a lot of Gouda cheese there, or Gouda cheese, depending on how you want to pronounce it. And that is a cheese that's susceptible to this spoilage. They actually have routine testing for these organisms, and they have a penalty system that they use to keep the level of these bacteria quite low in the milk that's going to be used for cheese making. Here are some, just a few additional photos of the defect, so you can see that they really have quite a lot of gas production in this defect. And then the last group of spore-forming bacteria that I want to talk about are what we call mesophilic and thermophilic spore-forming bacteria, and those are the ones that prefer to grow at sort of mid range temperatures, ambient temperature and above, and then even very, very high temperatures for thermophilic spore formers. And these organisms are used as specification tests for infant formula and for dairy powders and even for UHT milk and dairy products. So these organisms are present in the raw milk just like the others, but in contrast, we can also find these spore formers as in-plant contaminants. So if we're making dairy powders, we have to be concerned about the mesophilic and thermophilic spore formers that are coming in the raw milk, but then we also have to be concerned about the mesophilic and thermophilic spore formers that might be in the processing facility and contaminating the powder in the processing facility. So this one is a little bit different than the others, because of the others, we don't see them. The raw milk is really the primary source of those spore formers. And you can see I have some examples of specifications for exports of these types of products. And one of the reasons that, we're concerned with exports in this project was because there is an opportunity for high value exports of organic dairy products to countries, like the Middle East and North Africa, or even into Europe or into China. So these are organisms that we obviously have to be pretty careful about from the perspective of making sure we have very low levels of these spores in the initial dairy product, and then also in the end product for powders and for infant formulas and for UHT products. And so you can see the mesophilic and thermophilic aerobic spores here, quite low, less than 500 to less than 1,000 per gram. And then down here as well, the aerobic spores less than 500 to less than 2,000. Okay, so one of the big questions that we asked in this project was, since spores are found in the natural environment, and obviously dairy farms are natural environments, what factors affect the ability of those spores to get from the natural environment into the organic bulk tank milk? So we did this national study. And I'll run you through a little bit of what we did, but what we know about the practices and the management impacting spores overall and the overall milk quality here is that this is not a one factor issue, right? We can't just, for example, change the cow's environment or the bedding that the animal uses during confinement, or whatever, teat dip. You can't change one thing and expect it to solve this problem. It really is a very systems based issue where one thing can affect another thing, which can affect another thing. So we knew going in that we were going to be looking at a variety of different factors that would impact the level of these spore forming bacteria in the raw milk. So we developed a survey to understand, what are these farms doing? What practices are they implementing every single time that we take a sample, so we can understand what are the factors that are the most important out of all of these different things that happen on a farm. So we asked about a number of different things, and they fell into generally these categories of sort of housing and bedding management, environmental factors, milking system management, udder and teat health condition, and mastitis management. And so we did this study to understand, how do we prioritize these factors as far as reducing the spore levels in bulk tank milk. So we have we did testing on spore levels. We had just over 100 farms in this study. And we tested spore levels as well as total bacteria count every other month for a year. So we had 6 milk samples from each of these farms. We did a farm management survey at each of the time points that we collected a milk sample, because some of the factors change, right? We wanted to know things like how many hours a day are the animals on pasture at this particular time, and obviously that's going to be different depending on when we took the sample. And then we also wanted to know about meteorological conditions at the time of the sample collection, because we expect, or our hypothesis was, that because the animals are on pasture, that something like rain or humidity or temperature might actually have a bigger impact for these transmission of these spores from soil, which is their natural environment, to the animal herself. So all of those things combined, we used to analyze to understand what's driving the level of these spores in the organic bulk tank milk. So like I said, we had a little over 102 certified organic raw milk producers enrolled in this study across a number of different states. This was a national study, and we had collaborators in Pennsylvania, in Nebraska, in Oregon, and in California. And working together, we were able to collect these samples every other month for a year. And all of those samples came back to my lab at Cornell, and we analyzed them for these different spore types that I talked about earlier, as well as the total bacteria count, because we wanted to use that as sort of an overall assessment of the quality of the milk, because that's a measure that we are very familiar with, and we have a lot of more data on what is the level of total bacteria in the sample, as opposed to just the level of spores. As far as the survey data, like I said, we collected a lot of different data from these farms. We asked them a lot of questions, and they were all very patient with us as we asked these questions. But we wanted to know, some general farm information. We wanted to know about what they were fed, both when they were on pasture as well as when they were in confinement. We asked a lot of questions about management, and especially parlor management, and then the milking practices as well. And then, like I said, we gathered all this meteorological data, which included air temperature, humidity, precipitation, wind speed and wind gusts, and solar radiation. There are other studies that we were kind of relying on to understand how these parameters might impact the transmission of these bacteria, or the transfer of them from their environment onto the animal herself. And so we used some of that existing information to identify what parameters did we want to study here. And then, because we had a lot of meteorological data, what we did is a technique called principal component extraction. And I won't go too much into the technical detail here, but just a factor is that, a lot of times these different parameters are related. We wanted to distill all of this data that we collected, hundreds of thousands of data points. We wanted to distill this down into a few factors that we could use to determine what's really driving the impact of the meteorological conditions on the spore level. And so we took all this data, we did a principal component extraction, which essentially just allows us to say, okay, here are the variables that are similar to each other, and they kind of, they're associated with each other in a way that if, the temperature one day before the sample point that we collected is very, very similar to the temperature 2 days before the sample was collected, right? And so those are kind of all lumped together. So I bring this up not because I want you to understand principal component extraction, but because when I get to the results, you're going to see some principal components, and I want you to understand what that is. So that's just these meteorological conditions, right? So then, just overall, what is, the level of these bacteria that we found? That was sort of the first thing that we wanted to understand, because we want to be able to compare that to, other things that we know about spores in bulk tank milk, right? So you'll see here that we have these different titles, and I'm just going to remind you what those mean, even though, we kind of talked a little bit about them. So these are those anaerobic butyric acid bacteria, that's BAB. Here are our mesophilic spores, our psychotolerant spores, and our thermophilic spores. So you can kind of get the sense of what these are. So on those anaerobic ones, we know we need as few as, 10 per liter can cause that defect. Typically, it's a little bit higher than that, 100 per liter or even 1,000 per liter. We had a mean or an average of 148 per liter of these spores. The MPN is just the method that we did. You don't have to worry too much about that. You can kind of interpret it as spores per liter. For the mesophilic spores, very low levels, which is what we expected, because these do tend to be in milk at very low levels. Less than 6 spores per milliliter. So very, very low. If you're familiar with the a total bacteria count. Typically, we're talking in the thousands, right, of bacteria per milliliter. And here we're talking less than 10. Same thing with these thermophilic spores. So a little less than 3. This actually should be CFU per mil as well. So spores per mil. And then for the cyclotolerant spores, again, we're looking at per liter, not per mil. So only 31 spores per liter. And now keep in mind, this is enough to cause spoilage in dairy products. They're very, very low levels, but we can have very low levels and then they grow, eventually spoiling the product toward the end of shelf life. So we put all of this information together and we used a machine learning approach to analyze this data. And that is an artificial intelligence approach. Essentially, what we did is we trained a model to understand our data set, and then we allowed it to tell us what are the patterns in this data. And this is a really useful way of using machine learning, is just to analyze data, right? So we're not doing anything spectacularly fancy with this, but the advantage that it gives us, and the reason that we like using it for a study like this, is because we have so much data. We have all this survey data. We have all these principal components from the meteorological data, and then we have all of this microbial data, right? And we can put it all together and we can analyze the data together. In traditional statistical approaches, you can't do that. And so you get biases in the outcome of your analysis because you are having to do some initial selection of factors that we don't have to do with this approach. So all of that to say, feel how you will about artificial intelligence, but it's a really interesting way of analyzing data, and a very effective way of analyzing data. So let me walk you through this because it's kind of a busy plot that we have here, but I think we'll take a minute and break it down. So what we did with this data set is we had a number of different types of farms, right? We had over 100 farms. We had a lot of different practices, and sometimes when you try to lump everybody together, you don't actually get the best viewpoint into what's happening on similar farms. So we broke this data down by farms that use parlors and farms that don't use parlors. We broke the data down by samples that were taken when cows are on pasture and samples that are taken when cows are not on pasture. And then we also broke it down by samples that were taken from farms that were certified for less than 9 years and samples that were taken from farms that had been certified for 9 or more years. And the reason that we did that one in particular was because when we did the entire analysis of the full data set, which is actually represented here in this first column, the amount of time that the farm had been certified showed up as a very important factor for the overall analysis. So that was something we weren't really expecting, to be honest with you. So we wanted to learn more about that, and the way that we did that was then to ask sort of the question, okay, what's different on these farms versus those farms, right? The more recently certified versus the ones that had been certified for longer. And I will come back to that in a minute, but we're digging into that particular factor a little bit more because our survey was obviously not designed to learn about certification, the differences in these farms based on certification year. So I will come back to that in just a second. So anyway, here at the bottom, we have these different subsets of our data. On the side here, we have all the different variables that went into our model. The number of cows, the certification year, the number of people working per week, whether the udders were clipped and flamed. And now I'm finally getting to the point of the entire presentation, right? All the way down. You can see again some of those principal components associated with temperature, principal components associated with precipitation, right? So we have all these different variables that were fed into this model. And then we have different colors. The different colors are those 4 different spore tests that we evaluated. So again, the butyric acid, the psychotolerant, the mesophilic, and the thermophilic spores, okay? The darker color means that the variable was found to be associated or important for all 4 of those different spore types. If you have a white box here, like on these ones, then that variable was not found important for any of those spore types in that particular data set. So here again, all the data combined together. Here is the sort of outcome for just the parlor farms, just the farms that are not using parlors, which by the way, was a combination of robots and tie stall farms. So those 2 things were combined together. Samples that were taken when cows were on pasture, samples that were taken when cows were not on pasture, et cetera, et cetera. What I really want you to take away from this is we found a few variables that rose to the top as being important for all farm types, for all spore types, okay? And the one that's on the very top is the number of cows. And that's not because there's something magical with number of cows. It's because it tends to be that larger farms have more standardized procedures in place, and that can affect spore levels. That's our sort of, it's a proxy, I guess. So cow number tends to be a proxy for standardized procedures at the farm. Again, that certification year, that one was super important regardless of the scenario. We did have somewhere it only showed up for 3 spore types, but overall that tends to be a really important variable, and that's something that we want to learn more about. And so we actually have a survey. So if you're an organic producer, and you want to help us to understand a little bit more what could potentially be going into this and why there's this difference between farms that have been certified for longer and farms that have been certified for fewer number of years, you can take that survey and help us to develop some understanding of what could be happening here. We've done focus groups on this, and we're now doing this survey. And I think Jessica is going to go ahead and put that into the chat so you can grab the link for that and take that survey. It doesn't take a whole lot of time, but we are trying to figure out more why we see that as an important variable. Number of people per week tends to be very important, but this one was really driven by those robot farms. So farms that had no people milking because it was a robot actually had lower spore counts. And again, we don't know for sure because we haven't done the study to actually validate this result, right? But it would appear to be, again, that it's about consistency of application of the milking procedure. And with a robot, that tends to be pretty consistent across all cows, right? And then we get to clipping and flaming udders, right? So it showed up as important across most of the farms we were working with, and the different ways that we subsetted that data. So that's something that we wanted to take an extra step forward and understand, okay, our model, which is just a bunch of spore data, so microbiological data, and survey data, which tends to be really difficult to work with, right? Because you're asking people questions and you're not actually observing something yourself or testing something yourself. You're relying on people's memory and how well they answer the question. So we wanted to take one step further and say, okay, this showed up as important in this overall assessment of all of this data. Is it actually important? Can we actually show that there's a difference in the spore levels in milk from cows that have their udder hair versus cows that have their udder hair removed. So that was sort of the next step in our study was, can we actually intervene and reduce the number of spores in the organic bulk tank milk? So that was the question that we were going to ask. We also wanted to know, is there a difference in the powder? So if we have raw milk, can we see a difference in the level of spores? But then if we make a dairy product out of it, is there actually a difference in the spores in that dairy product as well? So we chose milk powder because, again, one of the big points of this study was to understand, can we make really high quality export powder for the international market as an opportunity for organic dairy? So we chose to make some milk powder out of this milk. So it was a really simple design. We enrolled 4 farms, which is not many farms, but we enrolled 4 farms. And we said, we want to come take a bulk tank sample for a week. 1 bulk tank sample for every load of milk for a week before we remove the udder hair through singeing. And I'll show you a video of how that's done. And we also want to collect a fairly large amount of milk, several liters of milk from the bulk tank on the day before we do that intervention, because we want to make some milk powder out of it, right? And we tested the spore level in the milk every time we collected that milk up to the point of the intervention. And then we made powder and we tested the spore level in that powder. And then we went in and we singed off the udder hair. I will mention that about 60% of the farms in our study did not remove udder hair, and about 40% did. So it was a fairly balanced number of farms that did and did not do this. And then after we singed the udder hair off, we went back and we collected another several liters of milk so we could make more milk powder. So we have a before udder hair removal and an after udder hair removal milk powder sample. And then we have, again, every load of milk for 1 week after. And we did this on the 4 farms and tested for the spores. So what did we find? Well, oh, actually, let me show you this video first. So hopefully this comes through. This is our, quick little, how do you singe udder hair video. And I guess this tells you the outcome. We did find lower spore levels, but I'll show you the actual data after this video. So we walked through just the basic steps, and this is actually a video from the study that I took while we were doing the intervention of our staff. And we had, 4 farms that had never singed the udder hair from their animals before. Some of them were a little hesitant about it because they were worried, but we, were able to walk through the procedure with them. We obviously want to be careful because we're working with an open flame. So we, kept a bucket of water in a wet towel in case, we had any flare ups or anything. And we had a fire extinguisher as well. So first of all, you want to remove all the debris from the outside. So anything that catches on fire. We use a commercial singer. These are just commercially available. And then, so this is actually a different, so we did this both in parlor and in tie stall. And you can see this one's in a parlor. This cow was ready to be milked, so she was bouncing around a little bit, but this is not painful. It does not, hurt the animal. If you've ever, have your arm hair singed off or something, that usually the first clue that you're having a problem or getting a little too close to a flame is the smell. It doesn't smell great, right? But the flame moves quick enough that it just singes off the hair, and it's not it's not painful to the animal. So here's the data that we found. Again, I'm just going to walk you through these acronyms so you remember. These are those butyric acid bacteria. The APC is the total count. We wanted to measure that as well. The mesophilic spores, the psychotolerant spores, and the thermophilic spores. The black is pre-intervention, so the milk that we collected before the intervention. The gray is after intervention. The little line in the center is the average, right? The mean. And you can see for some of these, like the total bacteria count, we really didn't see a change in. We also didn't see a change in those butyric acid bacteria, but we did see a reduction in the mesophilic spores and the thermophilic spores, and not so much on the psychotolerant spores. Now, if we had done this on more than 4 farms, we might have seen a difference in these things. But because we had such a small sample size, we were actually really excited to see that we did see a reduction in those mesophilic and thermophilic spores. Because again, we want to know, can we use this as an approach to improve milk quality? And the answer from a spore perspective, from a milk quality spore perspective, is yes. We can see a reduction in those mesophilic and thermophilic spores. And then on the powder side, so we went ahead, we have a pilot plant at Cornell, and we made powder from this milk before and after the intervention. And we only tested the mesophilic and the thermophilic spores in this case, because those are the 2 types that are monitored for in powder. And so you can see, again, we have the pre-intervention and post-intervention. So you saw a dramatic decline for the mesophilic spores pre and post-intervention, and a pretty good decline in the thermophilic spores after the intervention. And you can sort of imagine that the reason for this is because we had this drop in the raw milk, and you can see that here, right? We had a drop, kind of a moderate drop in spores there. But then once you concentrate that milk into a powder, you actually concentrate the spores as well. And so you can now see the drop a lot more dramatically in the powder than you could see in the raw milk itself. But this is really exciting to us because this is a simple procedure, right? This is a really quick, simple, low cost, low resource intervention that results in a really dramatic improvement in these spore levels, especially for these spore types that are important for, expanding an export market of U.S. organic milk. So big takeaways, these spores, they're in natural environments. They transfer into the raw milk, and they go on to cause this finished product quality issues and spoilage in dairy products. But we know that, there's a lot of things that go into controlling the transfer of these spores. And we talked about all those different factors, right? That can influence the level of these spores that end up getting into the bulk tank milk. But we can show that we can incrementally reduce the level of spores through very easy interventions like removing udder hair. And so that's really exciting to us. We can't, control the whole thing with udder hair removal, but we can make a dent in the level of spores. And then, we will continue working on this. What else can we do to reduce spores? Is it about, making sure that the management practices are consistently applied at the farm, that the employees all know how to, consistently apply those practices? Are there other factors? Again, the meteorological conditions. It's likely that when the cows are, out on pasture and it's raining, we have to be a little more careful about what happens in the parlor and cleaning and making sure that we apply those practices. So there's a lot here, but we're really encouraged that with a very simple intervention, we can see this reduction in spore level in the bulk tank milk. So I'm happy to take any questions that you guys have. Can't see the chat right now, but I'm going to go ahead and stop sharing my screen so that I can take a look at that. It's like we have a few. Oh, yeah, sorry about that. So the CFU, that stands for colony forming units. And essentially that's just how we how we visualize a bacterial cell on an agar plate, which is how we count it. Now, that other one I said is per liter. So some of the tests that we do are per milliliter. Some of the tests that we do are per liter. And you can imagine if we're, evaluating a liter, it's a very low level of bacteria in a full liter of milk, right? So in some cases it's CFU per mil. In some cases it's MPN, which stands for most probable number per liter. So those two were different. And it's just based on what level do we expect to have in the milk to begin with? Is it enough that we can test in a mill, or do we have to test a full, do we have to use a different method where we can test a liter? Right? So okay, so there's an additional question about growing the colonies. So the CFU per mil question, those are actually grown on a colony plate. The MPN per liter method, that's done in essentially in a broth tube. It's actually a milk media tube, and then we use a statistical method that allows us to infer how much was there to begin with in the full liter of milk. So they're just different methods that are based on, how many colonies do we, or how many spores do we expect to be in the milk to begin with? Oh, and Rachel already explained all that in the in the chat, but thank you. I appreciate that. Uh, and then Susan has another question about udder hair and the amount of butterfat. That's a really interesting point, and I have not heard that, Susan. If you want to unmute and tell us a little more about that, I'd be happy to um hear more about that, because I've never heard about the udder hair. I mean, certainly maybe there's, a genetic component, right, about the udder hair being on, animals that are more adapted to cold weather, and maybe there's like a butterfat connection with that. But that's interesting. Why don't you tell us about that?
Yeah, I don't know for well, I know for modern cattle, but certainly in the early part of the 1900s, there was some work done talking about the amount of hair on the udder, and interestingly enough, the amount of hair on the scrotum of males breeding bulls,
Okay.
then and how that correlated to butterfat. And there's also some really interesting old stuff like I want to say 1600s that was written
Hmm.
about cattle and butterfat and that sort of stuff
Okay.
based on hair swirl and hair patterns on the escutcheon of these animals. That's actually been captured and reprinted. But
Okay.
it's quite fascinating, and
That
it's
is
a
fascinating.
little tangent to our conversation, but it would be fun to look at and see if that, you know.
You know what I think is really interesting? So I have someone on staff who's constantly reading literature from, you know, 1500, 1600, 1700, 1800. Like he really digs deep, and it's really interesting to see how much stuff kind of comes back around eventually, that you don't think of, in sort of the modern system, but people have, sort of made really astute observations for a really long time. I will say that I do notice a big difference in udder hair, by, on different geographical locations. So a lot of times there's, and it certainly it makes a lot of sense, but more udder hair in colder climates and less udder hair when it's warm. So I think there's a lot to it, but in I've been to some farms where those
I
udders
think
are
we get
downright fuzzy,
I think it's interesting too, Nicole, what you say about the old stuff. We spend so much time, looking for modern papers, and there's nothing necessarily wrong with that, but we tend to just throw anything out, oh, well, that was only in 1962 or
Sure.
1943 or whatever. It's total crap, and it can't be true. And we see that. I see it a lot in management stuff and nutrition stuff and that sort of thing. I'm curious about the udder hair stuff too, whether it really is geographically approximate, which has not necessarily been my experience. I think the geographical approximation or the herd tendency could also be influenced just by their breeding
Yeah.
stuff too. They tend to breed certain lines of bulls or whatever. It's there's more we don't know than we know about it, but it's kind of an interesting thing. It can be a really fast way though to do selection of animals based on their phenotype for looking for some specific characters.
Yeah, for sure.
Butterfat.
I think you're
Particular.
absolutely right that that there's a lot we don't know. I think that I see both sides of the coin. I think there's a lot of folks who are like, well, we already know so much, and why are we doing more research? But the industry does change, right? And we have different expectations of the product. And I think it's important for us to continue to investigate, because there is there things do change over time. And as you say, I think there's definitely a genetic component to this, and certainly that's an important piece to understand. So I've found this to be really, really interesting, and I've also been really excited to work with producers of all different sizes and all different approaches to to their farm management. And that's been a really, really great way to get a good I think perspective on sort of what folks need, and what they are sort of looking for as far as support in their management. Well, thanks everybody for joining us today. And we're gonna, again, if you have time to take that survey, we'd be really appreciative of that. We'll continue to collect some responses through that, but happy to connect with folks after this as well. If you want my email, then please reach out. I'm always happy to connect with folks as well. We are working on a publication for this. We've actually got two publications in review right now on this work, and a third that we're writing up and should be submitted for publication this year. So it'll be coming out.
Thanks so much.
Yep. Thanks, everybody.